So I just want to, I mentioned, I was giving talk on the last meeting about Acholeplasma. I have few results that I just gonna kind of mention again, and essentially just kind of, hope, that maybe we can create a larger community working on this project. So Acholeplasma is very interesting. I actually started to work when I was at Venter Institute. It is also a molecule. It has similar properties to Mycoplasma, no cell wall, also like relatively small genome, but this one has a standard genetic code. So all the Mycoplasma that we are working on right now, they have different genetic code. So it would be nice to have at least one member with standard genetic code that we can create those synthetic cells, we can perform those genome transplantation and so on. So essentially that will we try to achieve here. Now, there were a number of genomes, so the first thing was, can we simply use the tools that were already created for Mycoplasma to start creating the cells? Now, many genomes were already cloning yeast, as we know, is the very first step of creating synthetic cells. But when it comes to genome transplantation, again, only the one with non-standard genetic code were performed so far. And we tried a few times to move the Acholeplasma genome into Capricolum, but that never worked. So probably, all those proteins, they wouldn't have the proper, I guess, stop codons and everything. They would create all kind of longer proteins. So that never worked. So essentially, right now, using the existing recipient cells for genome transplantation probably will not work for Acholeplasma. So we started this project. So after I left Venter Institute, I was kind of lucky that there was one student in my lab that was interested in doing some experiments with this species, which initially was really, really hard. It was colony would grow within like 2 weeks on the plates, very few genetic methods, but and everything we tried didn't work very well. So initially we identified 2 strains that potentially could be, so there's Acholeplasma leitlaui that could be used as a donor recipient, so we have that. And then we start working on genetic tools, and we're hoping that this is going to give us one day genome transplantation, and genome transplantation just using genome from bacteria to bacteria, or genomes that were cloned inside yeast. Now, one of those Acholeplasma species, the genome was already cloned inside yeast, so we know we can do that. We can clone whole genome in yeast, but the transplantation will be next. So here is just a brief overview of the 2 species that we are working with. One of them is the Acholeplasma leitlaui strain PGA8, and another one 8195. The main differences are really that one has more restriction systems present. So the 89, the PG8A, I was never able to transform any plasmid into it. It evolves resistance really, really quickly, probably due to the restriction systems and maybe secreted nucleases. So that was very difficult strain to work with. But recently we obtained another strain that is restriction minus and recombination minus, very similar genome, and we start using it as a to develop some of the tools. Kind of, the way to distinguish them as we're gonna continue going into genome transplantation, there is a visual aspect to it. The one of the strain is more kind of compact, more yellow when you have the palette, and we also develop quick screen with a multiplex PCR that we can quickly run on the donor and recipients. Now, here the first step that we want to do is, as Carol did and others, in order to develop the genome transplantation, it is good to optimize DNA transfer with plasmids. Now, there was a plasmid out there which is PNZ18 that was existing that we obtained that with neomycin selection. So we start using this plasmid, and in addition, we develop our plasmid with Acholeplasma origin of replication and slightly larger plasmid with tetracycline resistance. After trying some of the initial protocol, Daniel finally get few colonies with electroporation, but then he continued optimizing it, and now we can get, he was able to get quite a few colonies, with both plasmids. The smaller plasmid, he was getting more colonies. And you could recover those plasmids, and they were all very stable. So the two gels below just show that the plasmid can be recovered from Acholeplasma, digest perform, and they look all intact. So no major rearrangements here. So they were great. Now, we wanted to be able to deposit the plasmids to add genes, so we created also another version of the PNZ18 with yeast elements. So those two, the PAL1 and PNZ18, you'll be able to get them from AdGene. Now, since our plasmid movement by electroporation was good, but we still were kind of unhappy with the way, with the number of colonies. We wanted to increase the number of colonies as much as possible. So hopefully we would have a chance of, doing the whole genome transplantation. Acholeplasma genome is 1.5 megabase, which is about 50% larger than the mycoplasma. So, it may cause extra problems during the transplantation. So we want to optimize the plasmid transfer first. And Elibio asked the question, could we use electroporation for genome transplantation? And since so far we had electroporation working, that is one of the avenues. Now, Daniel, what he did, he started simply just transforming, having now two plasmids with two different selections. He would take the wild type 81 strain, transfer it, and then obtain the pool of transformants, and then repeat with a different plasmid with the PL1. Keep switching the selections, and you could also do the curing of the plasmid. But as you keep passing them, they eventually will get cured. We noticed that the larger plasmid was curing very quickly. The PNZ18 would remain sometimes after definitely more than 50 generations. There was a large population of them still carrying it. But anyways, after going through those 4 rounds, Daniel plated the colonies and pick up some individual colonies just to see if any of those colonies are better. So it was only 5 transfer, which was not that many, 5 different electroporation. And we didn't do any mutagenesis here. It was just the natural ability to, or the ability of Acholeplasma to introduce random mutations. And here is what happened. When he selected on about 20 colonies, we noticed that there was half of the colonies had this opaque phenotype, and some of them had more clear phenotype. And essentially, all of the ones that had this opaque phenotype, they start giving us better results for transformation. Here is just example of one that we pick up, and now you can see the wild type and the evolved one. Using the larger plasmid with tetracycline resistance, huge improvement, and the same thing was for the smaller plasmid. Approximately, it is about 500 times improvement, and we've done it over and over and over. This is based, I think, on 3 or 4 biological replicas. We should have the arrow bars here. So that was quite amazing. Now, there was in the past, it wasn't that this is the first example. Other people in the past also were able to develop better transformation strain using such an approach, but that strain seems to be lost, and so it looks like we were able to recreate it for sure. So one thing that we also doing this right now, in addition to the electroporation, we are doing the same thing, evolving it in different conditions. So with PEG, different chemical methods, and we are hoping that we can achieve similar results. Now, we did attempted one genome transplantation by electroporation, but that was not successful yet. So we will see if, but that was just, one quick experiment. They never, conclude that this is just not going to work for sure. But just when you look at these results, now we are getting over 100,000 colonies in one experiment. And when we started with the project, we were getting few colonies. And those colonies can be now seen on plates within a couple days. So it is growing essentially as fast as the mycoplasma. Now, we did also notice that this Acholeplasma strain can adapt very quickly to many conditions. So when we first start essentially optimize the media, we use different incubators, and we notice that after a short while, you can start growing them in 30 or 32, 34 or 37. and they start growing very well at all those conditions. We didn't push any upper or lower limits. We didn't try that, but again, this shows that Acholeplasma can evolve to many conditions. Now, as I mentioned earlier, we have deposited those plasmids to add gene. You can get them. We also deposited the strain, or just shipped this week, I think, this improved Acholeplasma strain, which is going to be called DNE, to ATCC. So if anyone is interested of working with this for different purposes, maybe we want to make Acholeplasma that swims. Maybe that'll be useful. Whoever would like to use that strain, it's going to be there. A few other tools that were developed was transposon mutagenesis, and this is just the only time that we were able to transfer DNA construct to the PGA8 strain. We got only one colony, but we have a donor strain right now with tetracycline marker. The TN5 works better in the AD195 strain, but in this one, at least we were able to move the DNA. So we in theory have a donor strain right now. Now, since last meeting, we created this working group that has probably close to at least 10 members that are invited to participate. We had multiple meetings that trying to accelerate now. So having now good strain, having all the ways to transfer DNA, plasmid that can maintain, we can now start testing all kind of different things. So the group has couple goals here. And the main reason that I am also talking today is that I hope that maybe we can get one more member that would like to join us and do something fun with the Acholeplasma, because it's really nice organism, and we need definitely more help. So what we want to do, and some of this is already being started with the members of the working group, but a lot of work remains really to finish that. So strains with landing pads, so we can start installing large fragments, genome editing, all kind of CRISPR applications. Obviously, the main thing, genome transplantation. We would like to start creating hybrid strains. So hybrid strains by loading large pieces of DNA on a plasmid and inserting it or adding them to the landing pad. Now, Acholeplasma leitlaui is really closely related to phytoplasmas, which are plant pathogen and they cannot be propagated in the lab. So we are hoping that maybe Acholeplasma will be a model for phytoplasma and creating some kind of hybrid cells. Again, on a plasmid, we already can do that, but maybe we can fuse the genomes inside these and do transplantation. So that is I know that a number of people are interested in this. We would like to also turn Acholeplasma into organelle, so we can fuse it. So there is protocol of fusing whole cells with yeast, and that also works for those Acholeplasma. We can fuse them, and maybe we could turn it into organelle. Earlier I asked a question about, I'm almost done. I think that's the last slide. Just one thing is that, we mentioned that, I mentioned that maybe there is a problem with cloning large GC DNA in E. coli, and here is just an example. When you clone Mesoplasma genome in E. coli, as you're increasing the size to 30 kb, essentially you're getting no transformants in E. coli, but maybe that would be possible in Acholeplasma. All right, so just the very last thing is, if anyone want to join us, you are welcome to the Acholiverse. So if you want to participate in any aspect of that, just email me and I will add you to the group. So thank you, everyone. Thank you to the working group and all the members of my lab, especially Daniel for doing most of this work. And I will take any questions.
Nice job, Bogumil. Anyone? One of the appeals of this project is my ambition to, and others, to make transplantation work for non-Mycoplasma bacteria. And as you know, Bogumil, Carol Lartigue and Nasira Assad Garcia and I worked for years trying to develop transplantation for Mycoplasma genitalium. And so if we can figure out how to do, it would be great if I could transplant into E. coli, but maybe Acholeplasma is a better step. Are you concerned that Acholeplasma will just recombine in your selectable marker?
At the moment, not. And one of the reason is that we try to actually recombine plasmid inside the Acholeplasma. So we created essentially non-replicative plasmid with, I think, 500 base pairs and 1 kb homology regions. And so far we failed to get even a single colony. That would be the result of recombination and survival. So it seems also that it has very poor recombination, which is actually good for us for that other aspect of genome transplantation. So hopefully that is at least in this strain that we are working. So
And then, do you know if there is a calcium activated surface nuclease on the Acholeplasma surface?
I think you asked me that in the past, and I still haven't checked that.
Okay.
I still haven't checked that. So
because this is something that we know from Mycoplasma genitalium, that when you treat genitalium, and in fact most bacterial cells with calcium, that they have a nuclease, a DNase activity on the surface. And so if your donor genome is destroyed, transplantation is considerably harder.
Yes, a little bit harder, yes. Yeah, I mean, we are in the process of checking things. We definitely know that when you just mix plasmids or other things, they will destroy, mix with the cell, they will degrade it. So, but we have to kind of, I guess, still do a little bit more just to figure out what is what and what is happening there. But we're improving the plasmid transfer, which was the first step in Carol's work. So we're hoping that this will be this is going to lead us to the final goal of transplantation.